Today we are going to have a discussion on the relevance of drug discovery programs and eradication of tuberculosis. For this we have with us today Dr. Balganesh, who is the research director for AstraZeneca India Limited, who has been involved in developing the drug discovery programs, both directing and actively involved in participating in the research. Dr. Balganesh is a renowned scientist, is also in charge of this program for quite a few years. Dr. Balganesh, with reference to the tuberculosis as it stands in this country and worldwide, particularly also with HIV and the tuberculosis that we find around, what is the status of tuberculosis vis-a-vis its control and the present problems?
Tuberculosis is a very old disease. We have detected it even in mummies, bone TB, using PCR. The present scenario with TB is really frightening, and that is why WHO in 1995 declared a global emergency for TB. The statistics are difficult, but in terms of accuracy, but in terms of what the ballpark figures are, today we estimate that we have got greater than 16 million cases of TB worldwide. We get about 3 million deaths per year and about 8 million new cases every year. Now, TB and its spread has been really attenuated, the spread accelerated by HIV. HIV has made diagnosis difficult. HIV has also made the treatment regimen for TB much more difficult. So, effectively answering your question, taking these statistics together, it is a frightening situation. In fact, WHO estimates today that 1/3 of the world population has been exposed to Micobacterium tuberculosis. And if you just think about that, we are talking about 2 billion people.
But then what is the status with reference to, say, available drugs, drug therapy and their efficacy in general for the entire tuberculosis?
Right. I think if you look at why we are in this situation, that will answer your question of the drugs. Today's TB therapy consists of a 6-month treatment. I can challenge you, when the last time you got a sore throat, you were given an antibiotic to take 3 times a day for 5 days, you stopped it after the 3rd day. We all do, right? We need to take this drug for 6 months. So, compliance is the major issue. Apart from that, these drugs, none of these drugs are actually what we call as really FDA compatible today. The last TB drug, rifampicin, was discovered in 1960. So, we have had no new drug which is brought in the regimen for treatment of TB since 1960. So, these drugs have all severe side effects. No wonder compliance is an issue.
Does it also involve any financial aspects, or this availability of these drugs, or is also a question of only the kind of regimen to be followed as a routine which creates the problems?
The availability is not an issue. I think there is enough TB control programs which have made the availability of these drugs pretty easy, both in terms of cost. In fact, we call today the Koch treatment is technically free in this country. If you go to the health centers like the National Tuberculosis Institute or some of their health centers, the treatment is free. But 1, the patient needs to go, and 2, the patient needs to take it, and we have no control on that. And that is why compliance is the major issue in terms of TB is concerned. Yes, we have an issue that we have multiple drug resistant TB, which is about 1%, but it is still not blown out of proportion. That is why today, if we have a good compliance, we will have a good cure rate.
Does it mean then that we do not really need new drugs, or if we do, it is the new drugs and the way we have the regimen for treatment that we have to manage?
Frankly, with no drug which requires a compliance of 6 months with the amount of side effects is going to be acceptable to the patient. We need drugs which will reduce this duration of therapy to obviously the most ideal situation would be, let's say, 2 weeks. But we can accept something at about 3 or 4 months treatment because we can still manage the distribution of these drugs. But to thus answering your question specifically, we need new drugs which will reduce the duration of therapy. That's one. We need new drugs because drug resistance is coming on. It's only a matter of time before they become the predominant issue. And so both these reasons clearly tell us that we need new drugs which will help us to manage the whole situation better.
But in this case of drug therapy and the way we want to control tuberculosis, is anything like you can have the endemic areas where you have the treatments all the time, so that you take care of the base level low and new occurrences as you find out, and could there be different drugs for this, or there could be a same kind of therapy or combinations that go?
Because of the pathogenesis of this disease, where you can have an individual who has an active infection or an individual who is carrying TB, he has a latent infection. Now, you can think of treating the active infection group with a specific set of drugs and the latent infection group with a specific set of drugs, because you can expect that in an endemic area everybody has latent infection.
Yeah.
Now, the problem arises when the latently infected individual moves to a non-endemic area. Then he introduces TB.
It actually becomes an agent for spreading or initiating the disease.
So what effectively then, in the case of a latent TB person, you need to give him or her drugs prophylactically. Now, the question arises, if 1/3 of the world population is infected, it is impossible to go and treat all these latent infection people. I mean, it's just too large a population. So you bring in another criteria, that those who have higher risk in terms of their lifestyle should get prophylactic treatment. In other words, if an individual has latent TB, which if you can come up with a good diagnostic, and has the chance and has the risk of getting HIV, we should treat that person with prophylactically. So that person's latent TB is gone, and even if he gets HIV, it doesn't become an issue. Second, we should be able to treat health workers dealing with HIV people, because if they are latently infected and they have this risk of being exposed to HIV individuals, then that group needs to be treated.
Let me ask you the same related question other way. Like, we know now certain areas where HIV occurrences are supposed to be very high.
Right.
Now, in such areas, will it be logical to have treatments given to or drug therapies given to people around them, even without having a diagnosed or detected tuberculosis earlier, as a protective thing, so that if they are protected from tuberculosis insurgence of any of that kind, or it's not exactly correct in terms of maintaining it, and how does one deal with such problems?
It's an issue which not everybody has thought of, including the regulatory bodies. Let's take the scenario which you have suggested, that in a highly HIV endemic area, we go and treat everybody for latent TB, even though there may not be a diagnostic. Now, the issue there again comes that is it a 1-day treatment, 2-day treatment, or a 6-month treatment. Now, if the prophylactic treatment is 2 years, then it's going to fail. It's only a matter of time. You can do whatever you want, it's going to fail. And so you need, again, you need effective treatment for treating those type of latent TB. And today we absolutely don't have anything which we believe hits specifically only latent TB. The other point which I want to make is not just HIV individuals. The scenario which we need to consider, if we believe this data that 1/3 of the world population is infected, we are all aging. The lifespan of all individuals generally is going up. Old age leads to immunosuppression. So latent TB people start getting TB.
Sure. Yeah.
Second, I think where the biggest issue coming up today is in organ transplant. Because during organ transplant, you need to do immunosuppression. And when you do immunosuppression, TB comes up.
The patient will show up suddenly.
Right. So these are real niches where you need to go and treat prophylactically. And remember, organ transplant is what's happening. Everybody is talking about in the next 15 years. We are all talking about
more often than earlier. Yes.
Whether it is just skin or whether it is liver, this is something we are all dealing with.
So these are the other problems which are coming up
Yes.
with maybe technology advances.
Absolutely.
But in general, what do you think about the existing, say, deficiencies in terms of the tuberculosis therapy that goes on with all the available infrastructure as we stand? What kind of deficiencies stand out, particularly with tuberculosis in question, unlike other diseases?
I think we can easily categorize them. The first is we need to look at the deficiency. Okay, let's look at its steps. First is these drugs have severe side effects. So that's an issue. Second, these drugs need to be given for 6 months.
The regimen.
It's absolutely, we cannot deal with that. 3, we need to give combination. It's not 1, we need to give 4. And anything beyond 1 leads to drug-drug interactions and various issues. And it is very difficult to get 2 drugs to have the same pharmacokinetic properties. So if one has to be taken after food and the other one is best to be taken before food, and what do? You make a compromise and give both of them together, because expecting the patient to take it twice is impossible. When you give it twice, that means something is already suboptimum.
Okay.
Okay. Then there is a possibility that the metabolites of one drug interacts with the metabolite of another drug. And the classical example we have today is protease inhibitors become less effective when you give rifabutin.
Because it inhibits actually the protease activity.
Exactly. And rifabutin increases the metabolic enzymes are induced which work on the protease inhibitor drugs. So the protease drug protease inhibitors which are used for HIV are ineffective.
And how much is additional complexity with reference to individual physiology?
Individual physiology is a major issue. We have seen that in the Chinese population, the pharmacokinetics of rifampicin was very different as compared to the European population, because many of these drugs interact with the lipids in our diet. So depending upon the lipids in the diet, the amount absorbed varies like anything.
The effective concentration in the body will vary depending upon the diet and the physiology.
Right. So these drugs, from a compliance, combination, side effects, and the fact that we need to give them all at the same time are the major deficiencies with the present therapy today, apart from drug resistance. I am not even talking about drug resistance.
Oh, so that's going to compound it further.
Yes, it only compounds.
But do you think that what are any new approaches people are trying to take?
I think there are major breakthroughs on the way, and this has largely been facilitated by the genome sequence. In 1997, late 1997, the sequence of Micobacterium tuberculosis became available. The entire genome sequence became available. And then in the last 3-4 years, we have the sequence of Mycobacterium leprae, we have the sequence of Mycobacterium bovis, and today we also are, it's nearly completed, the sequence of the vaccine, the BCG vaccine which was used, which has been used. This has actually put us in a position by using comparative genomics and asking the question, what is unique in Mycobacterium tuberculosis? Why does it divide only once in 24 hours while every other bacteria divides one in 60 minutes? How does it survive within the macrophage?
Inside the cell.
Inside the cell. How does it able to nullify the immune defenses of the host? So we know all that information is there in the genome. So now when we start comparing this genome sequence with other bacteria, we start unraveling what we call as virulence specific genes. Genes which contribute to the virulence of the organism or the ability of the organism or the microbe of the bug to cause disease.
So we are trying to get from the genomics the information which makes the mycobacterium cause the disease on one hand, but could it also help us find out the soft spots of mycobacterium which can be used for attack, and would it be for the new drugs possibility?
Absolutely, because I think if you are able to understand or get glimpses as to what is the process by which it evades the immune system,
the host immune system or the patient immune system.
Yes. Then if what you are saying is then it must be producing something which the host system then is turned off. Now, if we can block the production of that host, of that microbe factor, then you can enhance the ability of the immune system to eradicate them.
The patient will be able to tackle it
Absolutely.
without interference by the bacterium itself.
Right. That is one possibility. So, we are able to really, we can home in now on new targets, which we had no idea before.
When you say targets, it brings me the question, like people talk about discovering drugs, like I also asked you, but what exactly is discovery of a drug? Means what is meant by this discovery of drug, how difficult it is, and why people say drug discovery is not very cheap, is very costly, and so on.
Number one, drug discovery is really multidisciplinary process. It's a lengthy process taking about 8 to 10 years before a drug or a compound, a test compound from the laboratory bench is actually brought into clinical trials, followed by launching for use on humans. There are various phases. It's starting from the phase where we actually try to look at what we call as targets. Targets are nothing but functions or molecules which cause the function within a cell, so that either the microbe is able to breathe, multiply, or many metabolic processes. Any of them can be a target. Then we need to use that target and find compounds or chemical molecules which inhibit the function of them. That is the most preliminary step, and unfortunately our concept of drug discovery stops with that. But that is the first 10 to 20% of the entire discovery process. From here on, we start talking about using the chemical in animal models, able to show that it works in animal models, in infection models in the animal. We need to show that it is non-toxic. We need to show that it can be given orally. We need to show that it is distributed well in the body. We need to show that it is metabolized. We need to show that the metabolic products are not toxic. All that is done in the animal model. Then we need to take this molecule and do what is called as toxicology tests, where you treat one rodent and one non-rodent species for various periods of time with multiple doses, and analyze all the organs, analyze all the tissue fluids, both for concentrations and any changes from the normal pattern.
So, all these you are talking about the time span that requires is for a possible drug candidate to come to a visibility is about anywhere between 7 to 10 years?
More like between 10 to 12 years.
10 to 12 years.
What I spoke about up to doing the safety is about 4 years. After that is the clinical trials, which is the most lengthy and expensive portion of developing a drug, because in phase 1 you do safety studies on normal individuals, then phase 2A you do dose finding, phase 2B you do proof of concept, and then in phase 3 you go to clinical trials in terms of infected patients, then phase 4 you do blind trials and multicentric trials. That is another 4 to 5 year process. At each stage you develop you generate the data, you have to go to the regulatory agencies to produce the data, and they say, okay, the data are clean enough to go to the next step.
So, does it mean that in terms of the cost, for example, could you just give us an idea of what it will mean in terms of actual money that one needs to invest to possibly lead to a possible drug candidates?
Okay. We spoke about actually moving a molecule from one phase to another phase, but what we have to remember is that the chance of a molecule going from step 1 to step end is less than 10%. So, if I have to find a molecule for my tuberculosis, if I go through the process 10 times, I might be able to succeed 1, right. So, any cost factor builds in all the failures, because the final success of a compound has to build in all the failures that you have.
Right.
So, if you build in the failures and the success of a single compound, today it is anywhere in the region of $350 to $500 million, out of which 80% or maybe 70% is spent in the last 2/3 of the process.
So, it is really a costly affair to start with to get an effective molecule possibly, even before you then think of producing it on the large scale for actual distribution and all those. So, in that case, considering that kind of situation, where do we stand today? Considering our present problems of tuberculosis, the way we are facing it at this country, and vis-a-vis country like developing countries including India, which has more potentials to have infrastructural strengths.
Right. I think this is where we need to differentiate the basic scientist from the pharmaceutical person. Differentiate in the sense we have different ways of looking at it. I think as a basic researcher and a scientist, we need to take molecules up to the discovery stage, where we are mainly talking about in vitro as well as some amount of animal models, right. This is not expensive, this is less expensive, and it does not involve too much of technological issues here. It involves a lot of chemistry, but chemistry does not necessarily mean technology. Taking it beyond, I think today only large pharmaceuticals can do it. That is clearly one route. The second route, which is becoming more popular, which I think is, if you ask me, many pharmaceuticals in looking at new drugs for malaria or TB will follow this route, and that is what is called as a public-private partnership.
Okay.
There are at least two organizations today worldwide which are really putting this model into fact. The first one is called the Medicines for Malaria Venture, which is a Geneva-based organization, whose which is a body which is funded by World Bank, Bill Gates Foundation, WHO, and various other countries. All of them have put money into that, and the way they do it is that they look for molecules all around the world that the different basic researchers have developed or discovered, and then take it and develop it.
Okay.
So the developing cost, which is the main cost,
is contributed by or shared by these people.
And taken by the Medicines for Malaria Venture.
Same thing is also being done for tuberculosis.
TB, there is an organization called for Global Alliance for TB Drug Discovery, which is now it's an organization funded by Rockefeller, World Bank, Melinda and Gates Foundation mainly. They have put money together, which is now again doing the same thing. Basic researchers find molecules, they will take the molecules and develop it.
Okay.
So what happens then is the pharmaceuticals risk comes down, as well as the individual's risk also comes down.
So you think that in future, or at least through a variety of efforts both India and abroad, there is a possibility of some new drugs coming in for treatment of tuberculosis, and there is a possibility of being able to at least give a better kind of treatment to try to at least partly eradicate this problem or control tuberculosis in general?
I am very hopeful. The genomic era has given a lot of new opportunities. It is already reflected by the fact that the number of researchers in TB has gone up dramatically. Okay. It will happen.
So, to sum up, what is your feeling in terms of the possibilities and future about the new drugs, the control of tuberculosis, either in terms of stopping tuberculosis or eradicating it, and where do we stand?
I am very hopeful. With the genome information available and the tremendous interest in the research community, it is only a matter of time that we will discover new molecules, and with public-private partnerships available, we will succeed.
Oh, that is a good thing. Thank you very much, Dr. Balganesh.
You are most welcome.
Thank you.